Thank you very much, and thank you for inviting me to give a presentation on the program on ontologies for neural structures, which we abbreviate the PONS program. And we think PONS is very appropriate because PONS means bridge. And in fact, we consider this, just like we have for the digital atlasing, to be something that provides the way to translate amongst all the different things that we talk about in neuroscience. So this program also had a history. There was an initial workshop that occurred in 2007 that was led by Dr. Larry Swanson, of course one of the premier neuroanatomists working today. And the goal of that workshop was to agree on a general strategy for developing a systematic, useful, and scientifically appropriate framework for neuroanatomical nomenclature, essentially a universal vocabulary for describing the structural organization of the nervous system. We certainly know that neuroanatomy has a very, very rich history, and unfortunately, this was developed over the course of many hundreds of years, but not in a way that made it particularly computable. And in the modern information age, as we've already seen with ontologies and other things, it's all about how do we use these things in computability to help us advance our science and not get in the way of our science. So we know in computer science there's an information handling problem, and it basically says different groups of data gatherers develop their own idiosyncratic terms and concepts in terms which they represent information. And when we try to put this information together, we have to constantly resolve these conflicts, conceptual incompatibilities, and this happens over and over and over again. And if you take any territory in the brain, any one of those places that we saw in the voxome space, and you put a pointer in it, you can find many, many, many different things that people say about it. And the idea is that, you know, again, this is very useful. It's useful to have a rich language, but if we can't interoperate between these, it really limits what we can do with that information. And as you saw from Rob's talk, we can do a lot with genetic networks. We can do a lot now tying that to the anatomy, and we'd like to have a way of building anatomical nomenclatures that makes it a little easier. So I won't go too much into what an ontology is. You saw this yesterday. You saw it in Sten's slide. But again, the main purpose of ontology is to express human knowledge in a machine-readable form. But I do want to remind everybody it's a branch of philosophy, so you know, it's actually a very old field unto itself. And one of the challenges we had, which I talked a lot about yesterday, was how to take advantage of this branch of science without delving too deeply into a lot of the different issues that oftentimes are seen as hampering us, but turn it into something that's useful for us to use, essentially, again, to provide this sort of Rosetta Stone, this sort of translation service. So the PONS program was initiated in September 2008, based a lot on the recommendations of the workshop that Larry Swanson initiated. And as Jan said, there were 3 task forces that were established, one that was called the Structural Lexicon Task Force. So the whole thing about ontology is that it focuses on definitions, and so we wanted to have a basic vocabulary, a basic set of rules by which we defined neural structures. The second was the Neuronal Registry Task Force, led by Giorgio Ascoli. That had a couple of different mandates. One was that Nat Hines from the GenSat project recognized that with the new genetic techniques, with all of the different sort of technologies that are coming out, there's all kinds of new neurons, perhaps new neuron types. And it's been recognized a long time in biology how you actually name these things can bring some order for humans reviewing lists. It can also provide things that are more or less easily parsable by machines, for example. And so even though ontologists don't care about what things are called because we have to communicate with each other, neuroscientists very much care about what things are called. We've had enzyme naming, we've had various proposals for naming potassium channels and things, and the question was, could we do the same thing for neurons? The final task force was actually what we call now deployment and representation, and those are the hardcore ontologists that try to translate the products that come from the neuroscientists into something that is computable and logically consistent. So again, the Structural Lexicon Task Force is led by David Van Essen. Its main goal is to define brain structures across scales. We created something, which I'll show in a minute, called the NeuroLex Wiki, or the Structural Lexicon Wiki, to expose those products. But essentially, the task force has been trying to agree about what a defining criteria for a brain structure is. And this is any type of brain structure across scales. Again, providing some strategies for new nomenclatures. We recognize that every time somebody does a study on the brain, they find a new way to cut it up into different pieces, and we'd like to have a way of making sure that we can tie those efforts together, providing some order about how we name things to avoid the confusion that we have. Right now we have about 15 area ones, and we'd like to differentiate amongst those. And again, the means for relating nomenclatures, not stopping people from proposing them, but just providing some rules by which you should propose them. So essentially, what the Structural Lexicon Task Force is doing is it's providing the means to provide what is it. So at our last task force meeting, we said what we really need is, as Larry Swanson indicated in the task force, in the original workshop, what do we use to define structures? And if you look, there are things like parts of brain, parts of spinal cord, system, tract, parcellations, projections, ganglions. A lot of these terms have been used over the years, but nobody's ever really tried to define what they were in a rigorous way. So in order for us to take any brain structure that somebody proposes, the first thing we have to specify is what is it? Examples are, some of the examples are here. So we're providing those definitions in these common terms. The second thing we want to do is we want to say, where is it? So when we define a brain structure, typically the first thing we do is we say, well, it's a nucleus, it's a laminar structure, it's a white matter structure, and then the very next statement where we usually say, and it is part of X, and that part of serves to help us locate it. It essentially is the terminological standard that serves with the actual atlas representation, and that's very useful in several ways. First of all, it allows us to group brain structures together in useful divisions, but the second thing is, even though space is very, very powerful when we work within a specific species, when you try to use space to translate across species, it becomes a little bit more difficult. It's very difficult to take a rodent brain and merge that into a primate brain. So we recognize that we need some semantic structure for going back and forth between brains, and we believe that having the territories, these large territories, more or less point to the same subset of structures would be very, very useful, because when I say it's in the hindbrain in a primate and the hindbrain in a rodent, I should be able to point to roughly the same territory. The task force also felt that it was very important that the products of these things be exposed in a way that the community could easily use them and also comment, review, and add to them. Unlike the voxome space, where you're taking a rodent brain and you're translating amongst several different atlases, here the space is potentially infinite. How many terms do we need to be able to describe the nervous system? So the task force does not think for a minute that they're going to do all of this definition themselves. They're just going to provide the basic structures and rules by which that can be done. So we turn to wikis. Wikipedia, everybody's very familiar with, because they're very good community platforms, and the idea was that wikis make it very easy to find, so you don't have to load a tool that knows how to read a hierarchy like Protégé. It's set up for community review and contributions. But unlike Wikipedia, which is really designed to tell you everything interesting about something, this had to be a little bit more rigorous because we were focusing on definitions. So we used something called a semantic wiki, and we also implemented a few forms that essentially provide a structure to the type of information that you provide. So we have the standard set of properties that people required. We have the ability to put references in so that you can see where these things came from. So the idea is that each term would have a page. Each page would have its own unique identifier. And because it's so important to tie these structures to space, we've also been working with a tool that you saw referenced already called the Scalable Brain Atlas, which essentially lets us embed in a location for anything that comes from an atlas into the page so that you know where it is. And if you click on this, it's hot linked to a three-dimensional representation and brain browser tool. And this is being developed by Rembrandt Bakker in the Netherlands. The second task force is dealing with neurons, and as I said, they had their task was one, to talk about naming conventions for neurons, but the second thing is to talk about how do we define neurons. We heard Lee Hood this morning say it would be nice for us to be able to look at types. There's been long-standing arguments about what constitutes a type, and we realize mostly when we talk about neurons, what we do is we define them by a set of properties that they have. We define them by their morphology and what molecular constituents they have. We don't feel in the neuron task force force and the ontology task force that we are going to codify what these types are. We're going to instead provide a consistent structure by which one could reasonably ask what sets of neurons cluster together and which ones don't. So the idea here is that the structural lexicon will provide some of those building blocks. It will give you classes that describe types and parts of neurons and brain regions, molecules, and techniques. And the neuron registry will essentially be a database that builds upon these to describe properties that are associated with these classes. This was built, I should say, on some work that had already been underway by something called the Patilla group, who had gotten together in Patilla, Spain, to deal with the nomenclatures of features of GABAergic interneurons of the cerebral cortex. So we were able to build already on a community effort, again showing how INCF can facilitate. Some of the products that are starting to come out, and these are on the working pages, is the naming of neurons. Gordon Shepard and Giorgio Ascoli has proposed a naming convention, and they've already implemented it for a bunch of neurons that are available on the Neuralex wiki in case anybody's interested in it. There's also been a Curator Interface that was developed by David Hamilton, who's here and is outside, that has allowed people to start adding neurons, and it's defined a set of properties that are associated with the neurons. And one of the important things about this is that these properties are assigned not at the cell class level necessarily, but at the part of cell, so that you define a location for the cell soma, for the axon, the dendrite, and so on and so forth. So this has already begun, and the first products should start emerging from this task force. The third one, as I said, is representation and deployment. And its goal is essentially to provide the more formal structures that sit underneath this. It is also working to make sure that all of the ontologies that are developed and used by the Structural Lexicon and Neuron Registry Task Force are aligned with community efforts. We don't want to isolate neuroscience from the rest of the life sciences community. We want to integrate it. So we've been working with the cell ontology group, the gene ontology group, protein ontology group to help improve their neural content. These are providing the building blocks then for us to describe neurons and make some sense out of all of the different ways that we can relate neurons together. So essentially all of those things that you see in the user interface have been formalized so that they are in a machine readable definition. This is work that's going on by David Osumi Sutherland. There's been some very lively discussions about what the nature of a chemical synapse is, an electrical synapse, what are the fundamental properties that we need, and how do we define them. So the goal, again, of this is to make sure that when we build within a framework, all of the pieces interoperate. So we're providing the base building blocks. By doing this, we ensure that what we have are computable and we can use to answer very basic questions. For example, if you cut this out and you blend it up to do a microarray experiment, you might want to ask what things are found here. Some of those are cells, but some of those are part of cells. And because we are all building this within the same framework, it is very easy for us, in the context of a query, to pull out not only the neurons that we say are in the dentate gyrus molecular layer, but all of the axons, the dendrites, the parts of cells, the molecules. Essentially, it provides a computable structure for going back and forth across scales. So again, the goal of the PONS program is to provide means to communicate across nomenclatures, to communicate across species, to provide essentially a common structure onto which you can build various tools, essentially providing the building blocks so that these things, no matter what interface you put up, are all speaking the same language, and to integrate us with the life sciences so that this rich information is available to the entire community and not just to the neuroscientist. Thank you.
Thank you very much. And questions from Maryann? Yes, please.
So, Maryann, this is a fantastic process. I just wanted to get a feeling for how authoritative this is. In other words, you obviously have the NIH and the INCF and others behind you, but have you got all of IBRO and SFN and everybody else also aware of and backing this effort?
SFN has been a tough one. The NIF project actually grew out of an initiative of the Society for Neuroscience. Once the NIF initiative was launched, the Society has said, okay, we've done our part. I think that the idea of this sort of computable and basic building block ontology has not yet permeated into the Society's consciousness. What I said yesterday about rank and file neuroscientists is true. I mean, essentially they don't really care about this. But the nice thing is that these building blocks are there. They are aware of our efforts. Giorgio has been working in the context of EBRO and some of these other things, particularly to start harnessing activities in third world countries, and so we're hoping to bring them on board. We've tried to make this as global as possible. Obviously, in the beginning, people have to start somewhere, and then the idea of all of the INCF programs is that once you have a nucleus, once you have things, you need to turn this over to the community to comment, and I think that that's going to be happening in the next year. Okay. But if there are people we're missing, let us know, because we'd like to bring them in.